So, a very good morning, everyone. So, the topic which I have chosen today is entitled as maternal mast in pregnancy. What are its implications for the fetal and offspring health? So, my outline will be I'll first discuss what physiological adaptations happen in a normal pregnancy. Second, how these adaptations differ in maternal mast pregnancy. And then what are its impact? What do we know about its impact on the mother and the offspring? Some evidence from the human studies. And then lastly, we'll discuss the mechanistic studies and some of the work from our own lab. So if we look at maternal metabolism during pregnancy, we know it is very precisely timed. So it is divided into two broad phases, an early anabolic phase and a late uh, pregnancy or a catabolic phase. Now, during the early phase, the maternal or the mother's body prepares itself by more storage of nutrients so that it can be supplied to the fetus during the second stage. So, in the late stage, on the other hand, this storage phase is shifted to more of a catabolism phase, where all the organs they actually give rise to more nutrient supply to the fetus. So, if we look at different organs, so if we see the liver in the early phase, the muscles and the adipose, they are all insulin sensitive tissues. So the liver with increased insulin sensitivity leads to more glycogen deposition. The muscle, of course, takes up more glucose, more storage again in the form of glycogen. The adipose, again insulin sensitive, more of lipid storage. In the late pregnancy, there is a state of insulin resistance. The liver is now insulin resistant. It, instead of storing glycogen, it mobilizes the nutrients. It is more synthesizing glucose. So it is in a gluconeogenesis phase. In the muscle, the glucose transport decreases, and in the adipose tissue, the lipolysis increases again because of the insulin resistance. So all these are happening, but the pancreas in both these stages, it is synthesizing enough insulin so that there is no or less hyperinsulinemia in the mother. And these changes in the organs are actually endocrinally regulated by the placenta. Placental hormones like the lectrogen, estrogen, they all actually ensure that different organs, they work up in this regulated fashion. Now, what happens in mast cell? So we all know that mast cell, which was earlier known as NAFLD, this terminology changed in 2023, which now is defined as the presence of a hepatic steatosis, that is fat in the liver, and then any one of the cardiometabolic features. Now, why we should care about mast cell in pregnancy? So the prevalence of mast cell has increased, and similarly, the prevalence of mast cell in pregnancy has almost tripled. In fact, in many studies where cirrhosis has been studied in pregnancy, mast cell is the leading cause. But unfortunately, maternal hepatic health in pregnancy is often overlooked unless the overt disease, liver disease, is clinically suspected. So we still are not recognizing maternal mast cell enough. Now, how do we compare organ-specific changes in maternal mast cell in pregnancy? So if you look at the insulin resistance phase in a normal versus mast cell pregnancy, so women with pregnant women with mast cell, they already have a pre-existing dysregulated metabolic environment, which is characterized by increased triglycerides, there is already a systemic insulin resistance and a chronic low-grade inflammation. So the liver now is actually synthesizing more of glucose, so more of increased gluconeogenesis. There is hyperglycemia. The adipose tissue, you have more of lipolysis. In the muscle, there's again reduced glucose uptake. There's an impaired glycogen synthesis. And the pancreas, as I told you, is the pancreas, as I told you, normal pregnancy compensates for the hyperglycemia, but in a mast pregnancy, it is not able to compensate. In the initial phases, there is hyperinsulinemia, but subsequently, this can result in beta cell dysfunction and less control of glucose. So now, what do we know about the impact of maternal mast on the mother and the offspring? So we have some evidence coming from the human studies. I have used both NAFLD and MASLD studies here. So now the most important and the first big study on maternal NAFLD in pregnancy. This came from Sarkar et al. So this was a large retrospective study. So the study actually, recruited pregnant females and characterized them into MASL using International Classification of Disease Codes. And they saw the prevalence of MASL that was almost tripled from 2007 to 2016. But only 3% of these patients had documented MASL or NAFLD prior to pregnancy. Pre-existing metabolic complications like diabetes, obesity, dyslipidemia, hypertension, they were obviously present more in the mothers which were diagnosed as NAFLD. And both maternal and perinatal or the preterm birth, that is the risks to the fetus, risks to the offspring were noted in this cohort. So maternal risks included preeclampsia, eclampsia, postpartum bleeding, and in the fetal or the offspring risks, there was preterm birth. So another study, so this was not a retrospective, now this was a prospective study where they screened MASLD or NAFLD by liver ultrasound in US. So they reported prevalence of MASLD of around 19%, and it increased with obesity, that is females with more BMI had more prevalence of MASLD expected. Now this again, important study from Korea. So this study recruited more than 600 women, pregnant females, and they reported the prevalence of NAFLD was around 18 to 19%. And in this population, around 6% developed gestational diabetes also. And those who developed gestational diabetes had the higher prevalence of NAFLD. And in terms of the delivery outcome, of course, we know gestational diabetes is associated with large for gestational age neonates. So this was also observed in the study. And most importantly, the prevalence increased with increased steatosis grade. So the same cohort was used in another study where instead of GBM, they studied hypertension. So they used the same cohort and they saw how many of the females with NAFLD actually developed neonatal hypertension. So obviously the women who had NAFLD, they had higher degree of the women who developed the offspring hypertension or the pregnancy associated hypertension had a higher rate of fatty liver. And in terms of delivery outcome also, this pregnancy associated hypertension females that delivered earlier. So this next study, this is important, although this recruited only 57 pregnant females, but here the fat liver was measured by an MRI proton density fat fraction. And here they talked about gestational MASLD. In fact, all of the studies which I have discussed, they did not see MASLD pre-pregnancy, but during the pregnancy. So almost all of these cases are gestational MASLD. So here the gestational MASLD again was identified in 18%. And it was correlated with BMI, of course, more obesity. And importantly, the study did not find any correlation of more muscle in mother with the fetal liver fat. It was not different, but the fetal liver fat positively correlated with the total cholesterol and the AST levels of the mother. Two important observations that the study made, that there was a much higher proportion of women who developed MASLT post-pregnancy. So this was a longitudinal study. So they actually gave the first observation that many females who did not have MASLT before pregnancy or during gestation, they actually had a postpartum MASLT. Then another observation that this study made was that all the fetal fat, what they observed in the ultrasounds during the gestation, they actually were not present in the offspring at the follow-up. So they followed both the mothers and the offspring at 9 months of age. Then another important study, which was recently published in Hepatology International. So this again was done on a very large group of females. And this, in fact, the major shortcoming, this study I only reviewed, the major shortcoming of the study was that they used a non-invasive liver index, fatty liver index, to diagnose MASLT in this cohort. So, but they clearly showed that the MASLT was independently associated with the risk, twofold increased risk of adverse pregnancy outcomes. So it was higher risk of GDM, higher risk of hypertension, preterm births, and no birth baby in the infants. And the more the number of cardiometabolic risk factors, more were the adverse pregnancy outcomes. And they also recruited females which were having MET ALD and ALD, which also showed similar results. So they concluded that basically steatotic liver disease in presence of coexisting cardiometabolic abnormalities is an important predictor of the adverse pregnancy outcomes. Then this important study, I found it in a reverse manner. So there they did not check maternal MASLT, but they picked up all the children who had biopsy-proven MASLT, and retrospectively, they looked at their mothers, and again, they found that maternal obesity was a risk factor of NAFLD in the offspring, and almost threefold higher risk of NAFLD in the mothers who were obese. The same cohort, Swedish cohort, here they used biopsy-proven mothers, and they again concluded that maternal MASLT was associated with increased risk of preterm birth, higher cesarean deliveries, low birth weight, but no congenital malformation or neonatal deaths. And they did not find that this pregnancy risk was increasing with degree of steatosis. So that the degree of steatosis was not considered significant in this study. Now, how do maternal MASLT actually leads to fetal and offspring effects? So some data from mechanistic and experimental studies that we have. So we all know about developmental origins of health and disease. We know, along with the genetic factors, the maternal lifestyle factors during preconception, during pregnancy, during lactation, they have a major role to shape the lifelong metabolic health of the children. So these factors actually result in changes in metabolic signals in the mother, which can then lead to altered metabolic flux in the placenta and also in the maternal hormones. So all this can translate into change of the epigenetic processes. Like we have DNA methylation, we have histone modifications, we have non-coding RNAs, and these changes can then affect different physiological processes like cell proliferation, cell growth, and metabolic control. So what we see here is that placenta is an important interface between the mother and the fetus. And in fact, in normal physiology also, the placenta undergoes many changes in the early and the late phase. And the whole aim is to provide a balanced supply of nutrients to the growing fetus. But what happens during maternal metabolic dysfunction, what we know from obesity and from diabetic pregnancies, that there are architectural changes in the placenta. There's a lot of nutrient flux there's lipid accumulation also. The maternal microbiota derived metabolites also pass through placenta. The placenta shows inflammatory stress, oxidative stress, lot of apoptosis. The barrier thickness, the thickness increases, thus leading more to fetal hypoxia or hypoperfusion. Then the placenta we know is an endocrine organ. There's a dysregulated endocrine signaling in the placenta. And of course, the vascularization or the placental endothelial cells also undergo changes. So what happens in nasal placenta? So there are not many studies and it still needs to be identified. So all these adaptations in the placenta, they are poised to, support or ensure a proper or adequate fetal growth. And if you see all the organs, they start very early on in the embryo stage, but most of the development or maturation part takes in the later phase. So just coming to fetal liver. So we know that fetal liver development starts from week 5. And in the early phase, it is mostly, assuming hematopoietic functions. And at week 10, then it starts secreting bile. Then in the second trimester, the hematopoietic functions are at the peak and the metabolic functions come into play. It starts maturing in the third trimester. So most of the metabolic functions are actually in the liver are mostly at its peak in the early third trimester. So we can say that the fetal liver is a metabolic and hematopoietic hep during development. So the studies, most of the studies which have used fetal liver, of course, experimental studies, they have taken early third trimester, and they have been done either in the mice or in the Japanese monkeys or macaques in the third trimester. So this one report, which is interesting, this actually showed that the maternal Western diet exposure, which they gave for almost 5 years to the Japanese monkeys before they put them onto pregnancy, this increased glycolysis and decreased oxidative phosphorylation in the fetal liver and the bone marrow hematopoietic stem cells, both in the juvenile and in the fetal liver. So they showed both. This happens in utero and also in the juvenile or the offspring liver. So then they looked at the epigenetic changes. So there was an open chromatin conformation clearly seen in the genes which were controlling these metabolic pathways. There were also changes in the genes, epigenetic changes in the genes which were controlling inflammation. So this study, they concluded that actually these hematopoietic stem cells in the bone marrow and in the fetal liver, they are acting as a reservoir of the parent cells, and there is some intrinsic epigenetic reprogramming which is then passed to the fetus in utero and then to the offspring. Then next important change that is reported in utero is periportal fibrosis. So we all know that in pediatric medicine, it is most of the time periportal fibrosis as compared to the pericentral fibrosis, which is in the adults. So this periportal fibrosis actually starts developing in utero. So this study showed that again in the monkeys, if given fed on maternal Western diet, so there is increased activation of hepatic stellate cells as seen by the gene expression of SMA1 and TIMP1 and fibrosis. And importantly, the study showed in one of the experiments that even if you wean this monkey to a controlled diet, still this phenotype persists, which again says that the in utero programming is present in the offspring. The same group then extended this study to a more longer period. So here they used a 3-year juvenile non-human primate, which is almost equal to 9 years in the humans. So again, the study showed the similar results that a maternal Western diet, even if switched to a controlled diet or a Western diet, has increased periportal collagen deposition. And mechanistically, they identified a downregulation of some antioxidant signaling pathways, mitochondrial biogenesis, and upregulation of ER stress and fibrosis pathways. So again, the study pointed towards a perinatal origin for fibrosis and a longer term of disease pathology. Then this study used again the Japanese monkeys, and they showed that there is a altered bile acid regulation again starting in utero in the fetal liver, and an increased bile duct reaction, which is characterized by a CK9, increased CK19 staining. And this all starts beginning in utero. And they noted the similar changes in the offspring even after they were weaned to the control diet. So there is a dysregulated hepatic bile acid homeostasis inside the mother. Now this, again, a recent study, they showed the multi-generational effects of maternal obesity on mast cell. So here they studied the mice for 2 generations, and they showed that with generations, there is increase of hepatic steatosis. There is increase in fibrosis in the first generation. The fibrosis is there in the next generation, but it is less than the first. And importantly, the study showed that all this change in fibrosis is actually attributed to the worsening of mitochondrial function. So they saw mitochondrial biogenesis, the mitochondrial copies. So all those offsprings which showed increase in fibrosis had a decreased mitochondrial function. Importantly, this study showed that even if you don't feed the mother on Western diet, but just do knockout of the hepatic insulin receptor, that is, you induce hepatic insulin resistance, all these changes are still there, which again, focuses on the hepatic insulin resistance in mediating these kind of changes. So this recent study, so here they have used Kupffer cells in the liver. They have sorted the Kupffer cells, did some transcriptomic studies, and then they have used diets in the gestation, in the lactation, and the post-weaning. So in all the groups, so you have 2 groups where the mother is lean, and all the 4 groups are where the mother is obese. So even if there is no lactation Western diet, there is no diet of the post-weaning, only the mother's diet was enough to have a decrease in the oxidative phosphorylation G. If you see here, all the 4 groups had a decrease in the oxidative phosphorylation and increase in the glycolysis. So increase in glycolysis means increase in the pro-inflammatory signatures of the Kupffer cells. Now, in this study, they performed a single cell sequencing of the Kupffer cells also. So again, the lean mothers and the obese mothers, and they showed of all the subsets, of all the genes, there were 2 genes, ApoE and ApoA1, which was increased in maternal obesity in all these Kupffer cell subsets. And when actually these apolipoproteins were treated with hepatocytes, they led to increased hepatocyte lipid deposition, which showed a mechanistic link between Kupffer cell-derived paracrine signals and hepatic steatosis in maternal obesity. I'm not showing the full results. Another important point, if placenta is the switchboard, the fetal liver is actually the sensor of the maternal metabolic health. So what happens in a normal pregnancy? So in a normal pregnancy, all the blood which is going from the placenta to the fetus through the umbilical cord, umbilical vein. So this goes 60 to 80% goes to the fetal liver, and rest is then shunted through the ductus venosus. So this shunted liver blood then goes to other organs, and the blood which is gone to the liver, it is used in the liver, and then it is distributed to other organs as well. So this is the case of a normal pregnancy. But what happens in a diabetic or obese pregnancy? We know in diabetic pregnancy, there are changes in the placenta. There is a placenta hypovascularization. So, and we know there's hypoglycemia. So more nutrient is going to the liver. So liver needs more blood for the metabolic control functions. So more blood is diverted to the liver, and less blood goes to the other organs. So most of these babies have high adiposity, right? But most of these babies have shown changes in the brain and other organs. So this is when the liver gets priority. But in obese pregnancies, the studies have shown that there is increased barrier thickness in the placenta. So when there is increased barrier thickness, we know there is hypoperfusion. So less blood, the blood which is going to the liver is shunted most to the other organs. So this is called as a brain sparring response, so that these fetal organs are saved. Now what happens in mast cell? we can say that fetal liver is called as a metabolic brain of the child, but when this fetal liver is steatotic, how this metabolic brain differs, we don't know. So we do not know how relationship changes. Is it similar? Is it overlapping between obesity and diabetes? Between how much blood is going to the fetal liver and to other organs? We have no studies. But we do have studies, like I said, in diabetes and obesity, that these conditions in the mother lead to cardiovascular complications, neurodevelopment disorders, and even in utero, changes have been reported in the fetal brain and the heart. Now, coming to ILBS studies. So, we started these studies with Simran's PhD work. And the whole question was that pregnancies which are complicated by mast cell, what kind of intrauterine signals they have, which are, transferred from the placenta to the fetal liver, and finally to the other organs. So this is a part of funded DBT project. This is a brief study design where Simran is making different control and mast-celled animal models. So she's studying different organs in the fetus and as well as in the postnatal stage. And she'll also be looking at the functional assessment of other organs like the brain and the heart. And we also have planned a human cohort study for her thesis. So just to summarize her results in 1 or 2 slides. So she has successfully developed the mast-celled animal models in mice, and almost 70% of steatosis is seen in the Western style diet, which have been given both fat and sugar in the mothers, and there's considerable insulin resistance. There is increase in insulin resistance in normal pregnant females, like I already told you, but this is highly amplified in the mast-celled maternal models. And this shows hyperinsulinemia, and there's an increase in the lipid synthesis genes in the liver as well. And most importantly, these mice also show insulin resistance. Now, she's looking at the placenta as well. So in the mice, the placenta is divided into 3 zones histologically. It's a labyrinth zone, it's a junctional zone, and the decidua. So she is observing changes in all these zones. And increased angiogenesis or vascularization is also observed in the vasculature of the placenta. And we are also looking at the metabolic flux in the placenta, and these are few of the pathways which we have found enriched in the maternal mast cell placenta. And we see that amino acid metabolism, histidine, methionine metabolism, they were identified as most significantly enriched pathways, and a few metabolites going up and down. So currently we are understanding the relevance, and we are going to pick up a few metabolites for our studies next. Then she's also looking at the fetal liver, and the fetal liver also exhibits steatosis in the mast-celled mothers with increased insulin levels, decreased insulin resistance in the fetal livers as well, which again states that these metabolic changes, they start occurring in utero early. So then we have done a fetal liver transcriptomics, and we have identified inflammatory and stress gene pathways to be upregulated in the fetal liver. And some of the epigenetic modulators are also downregulated in our study. So again, we are trying to, connect links between the placenta and the fetal liver. Another student, Ahona, so the question for her work we asked was how this maternal mast cell pregnancy reprograms neonatal intestine development and the lymphatic lipid handling function, because our lab is looking at lymphatic vessels. So we wanted to see how this lymphatic development is reprogrammed in the neonates. So just showing some of our preliminary results. So the maternal Western diet is known to impair intestinal barrier integrity in the dams. It is also shown to have a compensatory epithelial proliferation in the maternal dams. And not only in the dams, we are also finding offspring changes in the intestine with decreased barrier permeability during the lactation phase, and also some changes in the proliferation. Again, what it means physiologically, we need to understand yet more, and studies are ongoing. So if I just conclude, the pregnancies with MASL, they are associated with adverse outcomes both in the mothers and in the infants. The presence of steatosis during pregnancy acts as a chronic stressor, which pathologically amplifies the gestational insulin resistance, leading to more of lipotoxicity and oxidative stress. And these disturbances then alter placental nutrient handling, the inflammatory signaling pathways, endocrine communication with the developing fetus. So the fetal liver is exposed to all these adverse metabolic changes at a critical window. And such early life exposures, it then leads to changes, especially in the Kupffer cells or in the mononuclear cells of the liver, which probably may play a critical role in the maladaptive repair or fibrotic responses, what we see in the fetal liver in utero. So what is the way forward? Firstly, we do not know what are the cutoff limits of identifying mast cell in pregnancy. So we need to define pre-pregnancy, we need to define pregnancy-specific criteria, the imaging thresholds. Like in a study for maternal mast cell, they use a threshold of 5% fat, but in the offspring, they used a threshold of 3%. So we need to define these criteria because we know that pregnancy itself is an insulin resistance phase. We need a long-term surveillance of liver in the high-risk mothers so that we can identify an early intervention window. We need longitudinal studies from starting from pre to during to postpartum changes in the mother, both in the mother and in the offspring. And of course, if we can create mother offspring cohorts, identify distinct metabotypes, because none of the studies which we discussed had included genetics in their analysis. So we need to include genetics, clinical and omics data sets to have more of these distinct metabotypes, high-risk metabotypes in pregnancy. And of course, we need to have more experimental studies where we can connect and, changes in the mother to the placenta to the fetus. So I acknowledge Sir for his inputs, Dr. Neha, who is helping us in all the understanding the placenta histology, Anupma for her contribution in optimizing the maternal mice models, Dr. Ankit, who is helping us in the human cohort studies, Dr. Jaswinder and National Liver Disease Biobank for the omics studies, the department, all my lab members, and of course the Center for Comparative Medicine. I'm grateful to all the collaborators, Dr. Picky from LHMC, Dr. Pooja and Dr. Monica from UCSF for their experts in the mast cell and pregnancy. And of course, I'm grateful to the reviewers because, so many critical comments we have addressed from this paper, which is currently under revision. And we are learning every day, and every day we are thinking that so little we know for the subject. And thank you all.
Thank you, Savneet, for such a lucid presentation. And I think there are challenges and it's a long way to go. Are there any comments or commenting?
Nothing we have done, still started.
Right.
For the fetal liver. How much data is there on this? And now we are reading it's so much, but all are recent. In fact, 2020 ke baad you will find many papers. So I think it is evolving, and but we don't know. So many things to do, and right time we started.
So in a way, it's the obesity which is more concerning here than
50% obesity, but
diabetes.
again they are saying whether it is an isolated muscle outcome, but or it is due to these cardiometabolic complications. We need to recruit, more patients, more screening, and in fact, when we talk to
So you have a human arm to it also, no?
we have put
some
So
part-
where are you taking those cases from?
So we are taking it from LHMC. We are getting those cases from LHMC.
And you're getting the placenta also.
Yes, placenta. All term placentas we are getting. So that's what when we are talking to the gynecologist, probably they're not looking at the liver during pregnancy, during the ultrasound. So
And you have a fair number of obese mothers there.
we have just started recruiting. So here our criteria is that we identify liver fat in ultrasound during pregnancy, in the first trimester. This is what we have started. Now, how many are obese? So probably that will depend on the numbers or the analysis when we do.
Any other comments? I know it's this proportion is definitely increasing over last 2 decades, obesity and pregnancy, and we are seeing much more as physicians getting more referral from. Of course, they're not looking for muscle only, but they're looking when they have gestational diabetes or they are having risk or they have hypothyroidism and all. So all that angle has to be seen. It's a good work, and I hope that one intervenes very early in pregnancy to offset the problem which may develop in the fetus. Any other comments? I think we just have one representation to do. Yeah, yeah. Pitching in. Thank you. Mr. May.